Hi everyone, my name is Stephen Lipscomb. I work for the University of Cumbria on their Back on Our Map or Boom project. I'd just like to say this isn't live. We had a few Zoom recording issues and we did actually lose the very end and the very beginning of the speakers. However, we did capture all that the speakers had to show. So all you did lose is a little bit of me talking about the Boom project and a little bit of an introduction to the webinar. So I'm just going to do a really quick intro now so you get an idea about the timings and when the speakers are going to be. And I have re-recorded a bit at the end where I talk around the Boom project, and then Michelle Cooper from Morecambe Bay Partnership talks a little bit about how you can get involved. So hopefully you haven't missed too much, and hopefully you'll enjoy what we have recorded and the speakers. It was a really enjoyable day and there's some really fascinating discussions. So thank you very much for taking the time to watch this, and I hope you enjoy. So just to orientate you through the webinar, I'm not going to bother with the housekeeping here now because it's not really relevant to those watching the recording. So first up, you will find me talking around the Back on Our Map project, giving you a bit of a background to it, what the project's hoping to achieve before we go straight into the speakers. First up, we'll have Naomi Dalton from National Trust. She will be accompanied by Craig McCoy, but he won't be doing any of the presentations, but he will be around for any questions and answers. Then we move on to Glenn Swainson from Natural England, again, accompanied by Ian Taylor from Natural England also. They have a break, I've edited that out, and then we will move on to Kew Gardens and Dr. Sarasan, who will be talking around orchid propagation, conservation around the world, and how the Boom Green-Winged Orchid project is feeding into larger research on that. Then we move on to my re-recorded bit on Boom, what we are doing with the orchids, what we're doing now, what we are doing in the future, and how we will need your help. Questions and answers haven't been recorded, but we will be hoping to put them out via a separate mail out or download later. And then finally, Michelle talks a little bit around how you can get involved and how you can sign up to be a volunteer or any other kind of contribution that you can give to Boom. So that's it from now. I'll move back to the live recording, and thank you very much for joining us. So moving on, Back on Our Map, Boom. So we've been going now for about 1 year. And as with all projects, we're working as part of a broader partnership. And as you can see here, we're working really closely with the Wildlife Trust, Morecambe Bay Partnership, and Natural England, among others. But really important to mention also is our funder. So like many projects working in natural environment conservation, we are funded by the Lottery Heritage Fund. We're very lucky that they've seen fit to support us in this work, as they do in many different ways in terms of natural and built heritage across the country. What are we doing? At the core of Back on Our Map or Boom, it's about working with communities. So it's about engaging with those communities and empowering those communities. And the method we've used to do that is around this idea of species restoration, and in particular, species translocation. And the underall aim behind all of this is this tagline here, and it's a biologically richer South Cumbria championed by communities. And that's really important, that the community is at the core of that. Who are we? So there's a team of 6 of us. We're all here, luckily. This hasn't happened very many times this year due to circumstances, so it's really lucky to have this picture that shows us all together. In the centre of this picture, we have Joe Sayers, who is the project manager. And if I go from the right, we then move to Michelle Cooper. Michelle Cooper is actually Morecambe Bay Partnership, and we've brought Michelle into the team to bring that expertise of herself, but also Morecambe Bay Partnership, that knowledge of working in the area and their community links already. And that's been really invaluable to us and really important to give us a flying start into working with these communities. And then going from the left, we have 4 project officers. On the second from the left there is Ellie Kent, Mick Mayhew, Deborah Brady, and then myself there on the right, Stephen Lipscomb. And between us, we're leading on the individual species translocation work. So where? Look at this map, it pretty much centres us around the Morecambe Bay area. But as you can see, there are fingers going up into parts of South Cumbria. So it is actually quite a large project area. Why? There's lots of reasons why you'd work in this area. And one of the main ones that keeps on coming up is the fact that this area is incredibly diverse in terms of its nature. It has been well recognized over the years by people coming here because of that reason. It's also incredibly diverse in terms of society and social factors though too. And this project has been centered here to try and bring those two things together into a cohesive means of giving a sense of place and a sense of location to this work.
Moving on from that, move to the second most important question is why. And I think the simple answer to why is just that why now is that now or never really. I think the data I'm about to show you, you've probably all seen in some form or another in recent years, but it's really worth repeating. And it's this idea or this fact that we've actually lost 68% of all biodiversity on Earth in the last 50 years. And that's a really humbling number. And that's across the planet. But if we come into the UK and break it down, many organisations are doing that for us. And the idea that we've lost 1 in 5 British birds in the last 50 years, as reported by RSPB. And the Mammal Society, that 1 in 5 British mammals face extinction in the next decade. And it really gives that sense of urgency and the fact that we really do need to be moving on these things now. Something else you've probably all seen as well, but really, again, important to mention, is this idea of the intactness of biodiversity. Out of 218 countries that this data has been collated for, where Britain lies on this is quite sobering, I think. And we are among the worst around the whole country, around the whole world. So really, it's just a matter of looking at the fact that we do a lot in this country for conservation, but it maybe isn't having the results that we need to do, that we need now. And so Boom is about looking at different ways of working, different ways of approaching this, and hopefully learning different things that can be added to the amazing things that are being done, but adding new facets and new methodologies to them. So I'm going to quickly go through the species. I said we are using species translocations as a means of sort of bringing communities together. And it's really important. I quickly just run through which species we're looking at and what we're doing with them. First on that list is aspen. We're going to be working with landowners and organisations around South Cumbria to plant thousands of aspen and try and link up the fragmented areas of these aspens that we do have in South Cumbria. It's a really important tree for sustaining invertebrates, and also really important in terms of sort of a wetland tree. So we're going to try and expand and link up those fragmented populations we have. The Duke of Burgundy, we're working with butterfly conservation to bring back butterfly conservation and organisations and groups who are already working in the area to bring back this really beautiful small butterfly by working on meta-population structure. Again, with butterfly conservation, but now moving over towards Barrow, we're doing work on the brownfield regeneration sites there to try and improve and extend that habitat to give this small butterfly room to expand its range. Goldilocks aster has a sort of unique place within the project in that it's probably the only species that is actually now extinct within South Cumbria. And that's happened really recently, probably within the last 20 years. So we'll be working to re-establish this species in the area using populations and knowledge from elsewhere in the country. Spike speedwell is the only scheduled plant that we're working with on the project. This beautiful charismatic plant, we do have one particularly good population in the region, and we'll be looking at what's working well for it there and using that knowledge and seed from there to see if we can work with landowners to expand the range of spike speedwell. Two sundew species here, great sundew and oblong-leaved sundew. These give us a chance to work with Cumbria Wildlife Trust. Now, Cumbria Wildlife Trust have done some incredible work in recent years, when I say recent years, over the last 2 decades, to really restore some of the boglands, lowland bogs that we have in South Cumbria, particularly around the Witherslack area. Now, these bogs now are ready to be plants that you'd associate with really good functioning bogs to be re-established. So we'll be working with Cumbria Wildlife Trust to translocate some of these plants onto these areas. Maidenhair fern is really working with a project that's been up and running in the area for a number of years now. A small group of individuals from the Arnside and AONB have been working to re-establish this fern at sites within the AONB. And we're really looking to work with them and build upon that to look wider within the area if there are suitable places where we can really expand the range of this plant. Hazel dormouse, the project's working with People's Trust for Endangered Species to reintroduce the dormouse to areas in South Cumbria. And this will be to reinforce an existing population in South Cumbria, and it will make it one of the furthest north reintroductions of this species in the country. Pine marten have been doing incredibly well in Scotland, and they've been expanding their range for a long time. The project will be looking at the feasibility and the appropriateness of bringing this animal back to South Cumbria. And then corncrakes we're using as an opportunity to work with communities and to look at the social impact of species loss and look at the community impact of losing these ubiquitous species from our landscape. And we'll be working with landowners and individuals in Rusland Valley and further afield to look at what it means to lose these animals and what it would mean to bring them back if we can. And then finally, but by no means least, the green-winged orchid. I'm not going to say too much about this now because we're about to pass over to many speakers who are going to talk about orchids and other things, but I will be coming back to you at the end to talk around green-winged orchid and what we've been doing with it. What I will say is that it really is partnership work again. We're working really closely with Kew on this to look at the scientific methodology that can be used to bring plants back and endangered plants back. And regionally, we're working with some of the local organisations, and we have this year Wildlife Trust, Natural England, National Trust, and Arnside and Silverdale AONB. And I think at this point, it's really important if we pass over to speakers from these organisations now who take you through what they've been doing to help boom, but also on a wider context in previous years to help wildlife in South Cumbria. First up, I think we're going to go over to Naomi Dalton from National Trust. Naomi, can you come back to us?
Yeah, just share my screen 1 sec. There we go. How's that look? Okay, hopefully everyone can hear me all right. A bit weird to be doing a presentation to the screen. I've just had a message saying my internet is unstable, so hopefully we last through this. So I'm Naomi Dalton, ecologist with the National Trust, working around the Morecambe Bay properties. It's a role that's also shared with the Arnside and Silverdale AONB, so I'm lucky to work on sites throughout this region. So just a short talk today, really, broadly around the green-winged orchid, but also leading that to grassland management and looking at some of the sites around Arnside and Silverdale that the National Trust looks after, how we look after them, and what the reason is for the conservation work. So lots of you already be familiar with the area, but just give it the basics to start with of where we are, what's here. These purple sites highlighted are National Trust property around Arnside and Silverdale. So you might recognise Eves Wood in the middle here, which is a large woodland complex. In the north, there's Arnside Knot and Heathwaite, which we'll be talking about in a bit, all the way down to Jack Scout Hill Brow in the south. These are predominantly kind of wood pasture, calcareous grassland, a nice mix of habitats, really rich in species. Some species like the green-winged orchid, a lot of other rarities, for example, the Teesdale violet and spiked speedwell, so important wildlife sites. We've got the fauna as well as the flora, so Scotch Argus here is holding on at Arnside Knot at the southernmost end of its range. And throughout the area, as with the rest of the AONB, kind of the underlying limestone and the geology of the area gives it its character and its special features as well. While there's a number of SSSIs, important conservation sites, National Trust also owns a farm in the middle of the village. This land here is a bankhouse farm, which is looked after by a tenant farmer. And that's where the good population of green-winged orchids are. So I joined the National Trust just a year ago, so I felt it was appropriate before going on with this talk to highlight the work of the people that are looking after the sites, to give them due credit. So we've got Martin Fishwick, who's the tenant farmer in Silverdale, and he really understands the sites and looks after them using his cattle as the main tool. So for example, at the lots. So it's Martin's years of work has made them what they are. Also the rangers, so here we've got Craig and Emma. Craig should be in the background today, so if you hear a voice button in, that's Craig helping me out, or he'll be around for questions at the end, hopefully. But Craig, Emma and Jamie are the three rangers for the National Trust who look after these sites. And we also have a very willing and keen team of volunteers who put in a lot of hours over many, many years when we just need lots of hands for the conservation work. So a good team of people working to look after these sites. Today I just wanted to pick out 3 kind of contrasting sites and take you on a bit of a virtual tour to them, looking at why they're special and how they're looked after. So we'll start off with the lots, which is in the middle here, before going up to Heathwaite. And if there's time, we'll have a chat about Jack Scout at the end as well. So starting off, the lots in Silverdale is a site I'm sure many of you will know, and many of you will try and make sure you walk through there in May when the greenwinged orchids are in flower, just a carpet of purple in one of the fields in particular. So I always find an aerial view is helpful to start with an understanding of a site. So these 2 fields here is the area we refer to as lots. You'll recognise this is Silverdale High Street on the edge of, on the right hand side of the screen, so very near the centre of the village. Looking at the lots from above, you can tell it's a picture's taken in summer, but it really picks out the areas with limestone protruding, for example, at the top of this field. Really thin soils. You can tell there's slightly thicker soils at the bottom of slopes where it's greener. But there's a lot of species-rich grassland throughout these fields, which the greenwinged orchid, for example, are one of the species that are found there. These fields are managed by the tenant farmer. So looking after him is very much down to his decisions on when to graze, and management of the fields. So there's no fertilizer or additions are putting onto the field, and cattle are grazed at various times throughout the year, depending on how it fits with his farm business as well, linking to the other fields he manages and has access to for his grazing. So it's part of the wider grazing scheme. But it works well, and his grazing scheme also takes into account what key times for particular species. So we have the green-winged orchid, which I think I'll just give it a brief introduction to the species. It's quite a charismatic species. Never mind the jester there. The Anacamptis morio. Morio is Latin for fool. So some people think this refers to the stripy sepals. So on the side of each flower, you'll see green veins, which are maybe the colour is reminiscent of a jester. Otherwise, the morio or the fool, maybe its features of deception. So even though it's got a nectar spur at the back of the flower, it doesn't actually produce any nectar. So there's no floral reward for any visiting pollinators. So they come in hope of a sugary feast and will pollinate in the meantime. But whether they visit another flower depends how determined they are. So the green-winged orchid in the lots grows in large numbers also alongside the early purple orchid. It's a species that was formerly widespread across the country, but has declined. A lot of decline is in abundance, so even though it's still present in a similar number of hectares, the actual number of sites within each hectare where it grows is much reduced. But doing well on the lots. So here's a graph of growth in the population basically over the past 10 years. So you can see we've got a willing volunteers. So one day each year, the volunteers will be out en masse on the lots, counting how many flower spikes they can find. And starting from maybe 1,000, and in 2019 they were counting over 7,000 flowers. So it's doing well, and doing well without too much kind of specific management input. There's no nutrient inputs, and it's unimproved short grassland, and the cows aren't on when they're in flower, but will be on other times of the year. So just doing well from a kind of low intensity grassland system. Also on the lots, benefiting from the kind of simplistic management is the autumn ladies tresses. You may know it from Jack Scout as well. And again, in recent years, it's grown amazingly in numbers. So that's a bit about the lots. Just a plain grassland field, which is very species rich. And in contrast to that, we'll head up to Heathwaite next, near Far Arnside. So Heathwaite is very much a habitat mosaic. You'll see here, surrounded by some mature woodland, and then with open grassland areas in the middle, with a lot of scrub mixed in there. So scrub of bramble, bracken, kind of hazel and ash and buckthorn, all creating a very dynamic system with lots of different niches for species to benefit from. Could be called maybe a wood pasture. There's areas such as these fields here which have very good priority habitat, grassland habitat. The objectives of management in a site like this are to maintain a mosaic, knocking back areas where the scrub is encroaching to make sure we keep good areas of grassland. And also ensuring there's connectivity. You may see this kind of track through here. The rangers will be working to make sure areas of grasslands are still linked up. So how do we do this? This is a lot of work for the rangers to keep the balance of grassland and scrub. Formally, it'd be grazed, so cattle do a bulk of the work. They would be on beginning of the spring and beginning of the spring and then beginning of the winter, kind of April and then September, October. So going through and shortening the vegetation, venturing into the scrub, but not there when at the sensitive times when the flowers are out. There's been increasingly feeling though that the cattle aren't really enough to keep the scrub under control. And bramble and bracken are increasingly encroaching into grassland areas. Whether this is a increased growth due to nitrogen, maybe atmospheric nitrogen, but it's reminiscent across a lot of grasslands in the areas that bramble seems to be increasingly a problem. So the rangers here have been trialling a few different methods, trying to find a combination that works to keep this mosaic, having open grassland areas which are species rich, having the scrub areas, but not letting the scrub overtake the grassland. So a few examples of the tactics here. In the middle, you can see a working party as a before and after. So this is a winter job for the volunteers. Selecting a different patch each year to extensively knock back the scrub, open up the grassland, and then let it grow again over the next few years. And by doing some rotation across the area, you have a mix of different ages and grassland reappearing where necessary. Other tactics in there as well is trying to beat back the bracken where it's smaller and encroaching in the areas. So the farmer has a bracken roller which he'll bring out, and that will knock back the bracken and create some dry litter for which is really good for things like fritillaries and some important butterflies that are found in Arnside knot. Otherwise, the rangers have a tractor with a flail. So this is summer work. So whereas the big scrub management is going on in winter, by more selective clearance, we can target bits of growth as they're appearing through the summer. So the rangers will be targeting areas with brush cutters as well. And they've been finding that rather than just a visit once, it needs a visit once or twice or 3 times in the summer to get on top of it. Repeating one area over the summer, and then maybe one more visit next year, will knock it back sufficiently for a few years and allow the grassland to re-establish and thrive. And also in a slide on conservation, you may be surprised to see a picture of the rangers in pesticide gear. So they're out target spraying some herbicide. So a bit of spot treatment of young bramble as it's emerging through the grassland, using Grazon, which is a non-persistent herbicide, and in very small quantities. They've found has actually helped keep the bramble under control and allow the grassland to persist. So there's a couple of trial plots set up on Heathwaite where they've been trying different methods, different combinations of timing of brush cutting, timing of herbicide, or just letting it be, and finding out that a combination of the methods helps decrease the bracken, lets the grasslands thrive, and also they're seeing the priority species within the grassland persist as well. But why do this? Why are we trying to maintain the grassland and knock back the bracken and keep that mosaic of habitats? It's full of mix of species that thrive there as well. You can see Craig and Emma here surveying the spiked speedwell. So Stephen mentioned the spiked speedwell, which is another boom species, and there's a population on Heathwaite, which has mixed success, but it's persisting, and we're hoping grassland management will help it thrive better. It's not just all the especially attractive ones that you might find this attractive. There's a dark bush cricket, one of only two populations of this species in the north. It's fairly common in the south and the Midlands. But it's clearly on at these two isolated sites in the north, of which Heathwaite is one. And this is a species that requires such a mosaic. So it needs the low-cut grassland and open sunny aspect to thrive for the year. But then its eggs are laid in shrub, kind of in bits of bark, where they overwinter for 2 years before hatching. So it needs the dynamic and the mosaic of habitats there, which we're trying to manage for. Okay, I think I'm nearly out of time. So I was going to cover a bit of Jack Scout. I'll just show a nice picture of Jack Scout because, again, this is another triple SI that the National Trust looks after. Again, working with a tenant farmer to graze here, and another site which will be hopefully hosting some of the reintroduced species through boom. Again, very important for orchids, in particular the autumn ladies tresses, and a few greenwood orchids here, but nowhere near as many in numbers. And again, it's a mosaic of habitats with areas of grassland, areas of scrub, some calcareous grass and some on thicker soils. And this site really shows the challenges of managing sites like these. It's a mosaic, so there's constant work to keep on top of the different scrub areas, letting the grassland thrive as well. It's grazed and it's part of the farming business for our tenant farmer. And then there's also a lot of visitor pressure, so you can see the paths cutting across it, which also can limit how much it can be grazed and what by. So there's a fine balancing act when looking after these sites and making sure it meets the best for the wildlife there, and also the use by people as well. It's a difficult site to manage. So that was a bit of a brief fly through some of the sites that National Trust looks after, and how we try and maintain a good balance of habitats for the different species there. Thank you for listening. I'll pass back to Stephen, and if you've got any questions, then myself and hopefully Craig as well will be around at the end for the Q&A session. Thank you.
That's great. Thank you so much, Naomi. That was really interesting. And as Naomi said, there any questions, if you can just pop them in the chat. We've had one come through already. Please feel free to add to that as we go along. I'm going to move on to Glenn Swainson now, who's joining us from Natural England. I forgot to mention before, but I will say that we do also have Ian Taylor from Natural England, who will be around possibly to help us answer some questions that you have later on. But before waste any more time, Glenn, are you ready?
I think so, yes. Do I just share my screen? That should.
Yep. Great. Thank you, Glenn.
It's better. Everyone see that? Okay, yeah. Good stuff. Well, my name is Glenn Swainson. I'm the Senior Reserve Manager at Gate Barrows National Nature Reserve, obviously in the heart of Arnside and Silverdale AONB. I'm going to talk to you a little bit today about the lady slipper orchid reintroduction that we've been part of on the reserve for over 20 years now. Just to put it into context, that's the blue line is the outline of Gate Barrows National Nature Reserve, probably a site that a lot of you are familiar with. Really diverse range of habitats. So primarily known for the limestone pavements at Gate Barrows, which are absolutely superb, and a really interesting range of vascular plants there. But it's got amazing limestone woodland. We've got large grazed areas, really interesting grassland, wetland, Haweswater Lake, obviously, and we've got an area of moss as well, Haweswater moss, which is fairly deep peat. So 120 hectares altogether, but a really, really diverse site. And that's sort of represented in the suite of different species that we've got. It's also a great orchid site. So I was just having a little tot up in my brain yesterday, thinking about the orchids that we've got. So we've got Northern Marsh, common spotted in the pastures. We've got a fair number of fragrant orchids, which weren't doing very well, but which we've done some restoration work on the mild grasslands around Haweswater, and we had about 12 spikes of fragrant this season, last season. We've got fly orchid, good numbers of dark red helleborine, especially around the pavements, broadleaf helleborine. I'm pretty sure we've got common twayblade. I feel like I've seen it, but I just can't remember where it was, but I know it's locally, it's quite common. We've obviously got lady slipper orchid, which I'm going to talk to you about a little bit more in a second. And then the possibility of a reintroduction of green-winged orchids as part of the Boom project, which is really exciting. So a really good orchid site, and we're very proud to have such a good suite of orchids on the reserve. So this is a plant that many of you might be familiar with, lady slipper orchid. It's one of the species, obviously, that Gate Barrows is really well known for, and we've been partners in the reintroduction program for a couple of decades now. It had the unenviable title of being Britain's rarest plant at one point, with only one known individual in the wild. But at sort of in the 90s, the Cyridedium committee decided that it wanted to do something about it. So in combination with Kew, they wanted to restore this plant to some of its former range. One of the issues with lady slipper orchids is obviously, like many orchid species, it's got tiny seeds, and it needs an association with the mycorrhizal fungi to germinate. One of the blockers was to figure out how to sort of germinate this plant effectively in the lab. So they were able to identify some individual plants that people had in collections, which were thought to be of native origin. Obviously collect some of the seed from the wild plant as well, and the cross-pollinated to create and fertilize seeds, which they then developed a technique to basically germinate those seeds in the lab without that mycorrhizal fungi. Once those plants were germinated, they were able to grow them on a bit for several years until they were big enough and robust enough to be able to plant out into appropriate habitat. As the Latin name suggests, they're found in limestone landscapes, and Gate Barrows was chosen as the sort of primary viewing site for this species and this reintroduction program. So they are planted out on a fair number of other sites in the north of England on limestone, but they needed a site where people could come and see them pretty easily, photograph them, and kind of have that experience with lady slipper orchid. So Gate Barrows was thought to be the perfect place. So we have quite a number of reintroduced plants there. The first batch went in 1995, and they all died pretty quickly. And one of the reasons for that is that slugs absolutely love we call them the noses, as they're kind of like pushing up through the earth early on. If they get eaten by slugs at that point, it really just does for them, and you don't get any flowering. So really pushed result for the first batch in 1995, but in subsequent batches from 1998 onwards, I think we sort of refined the technique of what care they needed to keep them going. So slug management was one of those things. A bit of watering when it got very dry, a little bit of weeding, and just to kind of a bit of gardening essentially to get them to flower. We also planted them across a variety of different habitats just to see where they would prefer to be, essentially. You know, I suppose if they died, then we knew that that wasn't going to be a very good site. Whereas if they persisted, then essentially we knew we were onto a bit of a winner. I suppose with all the care and attention that was lavished on the individuals that went in, it sort of masked that a little bit in terms of being able to tell which were good reintroduction sites and which were poor ones. And also because it became quite popular as a principal viewing site, it sort of developed an expectation that people would come at the end of May and get a really good show of blooming lady slipper orchids. So in some ways that put a bit of pressure on us, I think, to make sure that the plants were healthy and that they weren't being eaten and that they were surviving and blooming well. But it also meant that we weren't really able to tell which were the best sites in terms of where they were going to really thrive and do well, and where actually if we left them to their own devices, they wouldn't do very well at all. So in 2018, we decided that it was time to maybe let the plants, some of them that had been in the ground obviously for quite some time, we were going to let them stand on their own 2 feet and see whether they were going to thrive in those conditions or whether they were going to not do so well. What we didn't realize is that obviously in 2018 we had the hottest summer on record. So although we had a pretty good flowering period early on, it just got hotter and drier as the summer went on, and it really had quite an impact, especially on some of the orchids planted on the edge of the limestone pavement in an area of broken pavement. Just shift it on and just to get another image of those are some of the lady slipper orchids at Gate Barrows. So yeah, we had a really hot, dry year in 2018. As a consequence, 2018 was really poor. Hardly had any blooming orchids in the pavement location, which is the main area where visitors could see them. And then in 2020, this year, again, we had a really, really dry, hot spring and another very poor season. So it showed and demonstrated quite quickly to us that the pavement orchids, the main principal viewing site, probably wasn't going to be the most sustainable place for these plants longer term, especially with the forecasts around climate change. However, the pavement site wasn't the only site where we had planted lady slipper orchids across the reserve, and we'd also experimented with some lakeside locations next to Haweswater. And in both of those years that were very dry and very hot, lady slipper orchids really performed quite well, really nice blooms, weren't attacked by slugs, and we just had a good show. So it demonstrated, I think, that kind of level of care that we'd been giving the orchids in some ways wasn't the best thing, because we didn't really know where they wanted to be. And I think from our perspective, it feels like maybe some of these sort of slightly shadier, damper locations, especially around the lakeside, might be better locations going forward if we're going to reintroduce more lady slipper orchids. So that was quite an interesting sort of positive side to a fairly disastrous couple of years in terms of blooming lady slipper orchid. But then the other thing that we have to contend with is that the lady slipper orchids at Gate Barrows are all descended from the Kendall plant, which is one of the plants in captivity, which was subsequently shown to be of continental origin, so not strictly native. Other introduction sites that had used Kendall plants, they decided that actually there was a bit of a risk from outbreeding depression if they were left in those locations, especially if we were putting native provenance lady slipper orchids back out. So they were removed, that genetic material was removed from other reintroduction sites. But as all of the plants at Gate Barrows were Kendall or Kendall cross, it was decided that it maybe didn't matter too much. And actually it was that visitor experience that was the important part of Gate Barrows as a lady slipper site. So yeah, so that was a bit of an issue for us. I think from my perspective, I think we're now having the conversation about where do we go from here in terms of the reintroduction program. So Natural England and the reserve staff are really committed to be active partners in this program still. I think it's an amazing species, and we want to see that it reinstated back to its former extent and in the right kind of landscapes. Gate Barrows is definitely one of those sites where we feel like it should be. But we also kind of have to acknowledge that the lady slipper orchids that we've got at Gate Barrows are kind of non-native provenance from non-native stock. So And also we have to try and think about how we have a population of lady slipper orchids at Gate Barrows that are resilient to climate change. And it feels like to me that maybe some of the plants around and the broken pavement on the edge of the pavements at Gate Barrows probably won't persist of their own accord. So now it's just working with our partners and working with specialists, and obviously engaging with people who are interested in the populations to decide how we take it forward from here. So that's all I've got to say. Thank you very much.
Thank you, Glenn. That was really interesting. Yeah, and loads of really interesting questions posed there around the whole process that are really relevant to what we do, and a large part of why we're working closely with Natural England to try and learn as we move forward. But thank you, Glenn. That was really, really interesting.
Thank you.
I think I have us scheduled in now for a quick tea/coffee break. It was mostly to accommodate the chance that we might end up a little bit behind schedule. We're actually close to schedule, but I think it'll be a good opportunity for everyone to just gather themselves before we move over to hear from Kew Gardens. So if you want to just take a break, go and get yourselves a cup of tea, and we will reconvene if you can come back just before 2 o'clock, and we'll start sharply with Sarasan, Dr. Sarasan from Kew Gardens at 2 o'clock. Thank you, everyone.
Hi, Stephen.
Hi. I think we're just about on time. I think we're just before 2 o'clock.
Yeah, all went according to plan.
Yeah, really good. Really happy so far. Getting some good questions coming through as well. So
Very good.
encourage everyone to keep on thinking and keep on putting your questions in the chat, and we'll be directing these to the panel at the end during the question and answer session. But equally, I know that there are some incredibly knowledgeable people in our viewers, so feel free to chip in with relevant answers or furthering people's questions if you want. That'd be great.
Yeah. Sure.
I think we're just about at 2 o'clock, so I think if we share your screen, that would be a good place for us to start. If you're ready to go.
Yeah. Let me share the screen then. Okay. Okay. Is that okay? Great. Okay. Thank you, Stephen, for inviting me. And this is a great project to be part of. I was thinking about this presentation, how I should present this, because the work we managed to done on this project in the last few months because of the COVID was really challenging. So we managed to do some work, but at the same time, for this purpose, what I have done is I have put together some of the work which we have done in the last 10 years with the Natural England and the work done from Madagascar. So Orchidaceae as a family of more than 27,000 species. In the UK, about 58 taxa, and they are so diverse, and they occur these unique pollinators, the niche soil conditions, and the mycorrhizal fungus, as Glenn earlier mentioned with the lady slipper orchid, the problems we had, and in some cases, the continued reliance of the mycorrhizal fungus for further growth and colonization. So majority of orchids are found in the tropics, especially in biodiversity hotspots. So that is a serious sort of area of research when you go on to do some orchid work. So 10 years ago, I was thinking where will be the best place to go and work in orchids. And so at the time, one of my colleagues asked me whether we can produce, as we are producing the Cyridedium plant, so the lady slipper seedlings we are producing for Natural England, to do similar work in Madagascar. Madagascar is a haven for orchids. And at the time, I asked him whether he would be interested to do, in partnership with us, to do some work on the mycorrhizal, fungi and identifying them, because it was a long shot to even think about that, because at the time we didn't have much knowledge about whether this can be done or whether this will be feasible as a large project. But at the time, there was some support from Kew, and that, we managed to do quite a lot of work in the last 10 years. So as you can see, the global biodiversity hotspot areas got a lot of these orchids, which are so rare, unique, charismatic. So the terrestrial orchids are more dependent on mycorrhizal fungi compared to the epiphytes or lithophytes. And within that, the herbaceous terrestrials are more prone to decline in the wild due to the soil conditions or the lack of mycorrhiza in soil. Some of these are epiphytes, and some of them are lithophytes. On the right-hand side, what you're seeing is a terrestrial orchid. Orchids are so small that seeds are so tiny. On the left-hand side, we got the grass seeds. Grass seeds are so small, but just think about an orchid seed, which is so tiny. And when we look at them under the microscope, this is what you see. It hasn't got any food reserve. So all the food we eat as our staple, crops, the rice or wheat or whatever, actually the food stored for the embryo is what we eat. So these embryos don't have any food reserve at all. So what happens is the mycorrhizal fungus should support the early part of the germination process in all orchids. Further support may need in many other cases. So this is just an embryo. So there isn't anything else in there. This is the seed coat and the embryo. So when you look at a 1 pound coin, and 1 pence, I think it is, and then on the right hand side we got these seeds, and some of these epiphytic orchids from South America produce hundreds of thousands of seeds in a single seed pod. So they produce that many seeds for a reason, because only few of them will find the mycorrhizal fungus in the wild and then went on to become a seedling. Cyridedium, calceolus, this is the sort of seed capsule we get from natural ground volunteers. They collect and send to us. And this is a green seed pod. So hundreds of seeds, thousands basically. And then we grow them in a very fortified medium with all the nutrients the seeds need for its germination. So that means, what does the fungus do? What does the fungus give the plant is given inside a culture vessel. So that is one way of doing it. So you can collect the green capsule and then grow in culture, and you can produce thousands of seedlings. There is no problem. So in the wild, the orchid seeds needs mycorrhizal fungus, as I said, and some orchids need very specific mycorrhizal partners. That means the lineage of fungus is quite narrow. So you can't give any fungus. Say if it is a Ceratibacedium, a mycorrhizal fungus, if you got a Ceratibacedium and gave it to an orchid seed, it doesn't germinate. Even few base pairs different may not be enough. So that means it must be quite specific. In the wild, the mere presence of the mycorrhiza in the soil may not be enough. So mycorrhiza may be in the soil. Say, for example, the wild plant of Cypripedium calceolus may have the mycorrhiza around it, but there may not be enough mycorrhiza, in the soil to start the germination process. So when we see a field like this, there are so many orchids there. They are all flowering, healthy plants. But just to think about how many seeds these plants produce, millions. But in a year, we will see only very few seedlings in the wild. So there is serious problem with the recruitment of these orchids. I wouldn't say problem, but at the same time, if the density and diversity of the mycorrhiza is not that good for different orchids in a field, then it is a problem. So the things happened over the last hundreds of years. The land use changes, the soil characteristics must have changed over time, the nutrients, soil moisture, pH. So that means to study the way to find out what exactly these orchid seeds need for germination, we also need to understand the soil characteristics. So the climate change, Glenn said 2018 was a serious year for orchids. We had that, first-hand experience. We couldn't get any orchid seeds during that year for our in vitro work. Even if we got some seeds, they passed that stage. We always go for 45 to 60 days maturity because of the dry, long, period of dry conditions. They matured very early. And these are the reasons why, the temperatures and unseasonal wet periods, drought, they all can contribute to the colony decline of the mycorrhizae, both in distribution and their density. So if you get the proper seed at the right maturity, you can produce seedlings like this. This is Cyridedium calceolus. So I would say if you got 100,000 seeds, you can get 60,000 or 70,000 seedlings because the viability is so high. So it is feasible for any of these orchids for us to produce seedlings. But if you are asked, like the University of Cumbria in the early discussions, we were talking about the project, and then we went to this approach of identifying the mycorrhiza, then do symbiotic seed germination. Symbiotic means with the fungus, and asymbiotic without a fungus. And then we can reintroduce for translocation or assisted colonization, and then see how these asymbiotic or the seedlings without the mycorrhiza and the ones which can be produced with the mycorrhiza, when they are planted out in 5 years, 10 years time, is there any difference? So it will be a good starting point to study that sort of thing, because it has not been done in the UK. So we thought that will be a good approach. So to do that, we need to identify the mycorrhiza first, the standard, the conventional sort of approach is seed baiting. So you put some seeds in the wild, and after a few months, you may not get after a few months, but after a few years, you get the baited seed size seedlings. Then you can identify the germination specific fungus, mycorrhizal fungus. But for a project which is funded for 3 years or 4 years, we can't do that. So that means you have to go for spontaneous seedlings in the wild. That is also very difficult to sort of materialize. Then the only approach we can do is to get mature roots and then culture those roots and then do the DNA sequencing to find out what mycorrhiza is that. And then from that list of mycorrhiza we can find, then do the seed germination. So this way, the seedlings you produce will have the approach you will have lead to improved seed germination, faster seed germination and growth, and the better quality seedlings. I will show you some slides later how better they are. Then it will also enable seed storage at ultra-low temperature with the compatible fungus. Say, for example, if we identify the compatible mycorrhiza for green-winged orchid, and if we want to store it in a liquid nitrogen at -196 degrees centigrade, we can do that. That means in 100 years or something, if a species is extinct or very small number left in the wild, you can take out the mycorrhiza and the seeds and then start it over. So the symbiotic seedlings are important for natural recruitment of seedlings if you want to reintroduce back to the wild. That is a whole process we are talking about here. We need the symbiotic fungus. So I'm showing you some examples. The spontaneous seedlings we collected from Madagascar, we got, a tiny seed, protocome, which is only 2 to 3 millimeters. So as you can see, those lines are the sections we made, and from those we got the basal portion. If I can show you here, these basal portions are the ones where we found the mycorrhiza. You can see the colonization. As it goes up, the colonization of the mycorrhiza decreases. That means if you target this area, you will get the mycorrhiza. That has been, sort of the uniqueness of these, the protocomes or the seedlings you collect from the wild for any orchid. So they colonize in that sort of area, whether it is a seedling or a protocome. Protocome means the earliest germination stage of an orchid seed. It started germinating. That is where it is. Once you got if you can't get to the protocoms like the earliest stage, you can get tiny seedlings like this. Then you can slice them. You will get a section like this, and then you will get each cell colonized by this fungus. So basically, we can pick out each one of these and then culture. So this is a magnified view inside a single cell of this, the mycorrhizal colony. This is a super coiled mycorrhizal fungus inside a single cell. It fills the whole cell. And once you got that, what you got is you can culture it. And I said, so that is the approach. The second approach is, I'm going back to the slide. These are tiny seedlings. Then if you can't get tiny seedlings, what you will go for? This is the approach. You have to go for different sort of maturity levels. This one is a flowering orchid, as you can see the flowers here, but this one hasn't reached that stage. So any stage you can find, because that is easy to find. There may be more plants in the wild. So access and the diverse group of mycorrhiza you can find in root systems, because all these orchids recruit quite a lot of mycorrhiza during different stages of its growth, different seasons of the year. So the germination compatible fungi sometimes may not be there. If you are not lucky enough, then you don't find that mycorrhiza. That is a case I experienced with the case of Cyridedium calceolus. We identified other orchids from near the wild site, but we couldn't get these seed germination specific orchid. So as I said, inside the cell you got this super coiled fungus. You can take it out and put it in a medium. It will start growing like this. So it's explode within 48 hours or something in the best case, and then you can identify this fungus. So basically, we are hearing quite a lot about the PCR these days. The DNA is amplified, and then you do the sequencing. Once you've done the sequencing, you will know which fungus is which mycorrhiza, or is it a mycorrhiza or not. And if you got the mycorrhiza, and then you can culture it, as I said, you take a small blob of the mycorrhizal fungus, and then the seeds, you co-culture, then you will get these symbiotic seedlings. Basically, if you slice any of these seedlings, you will see this fungus here in the agar block colonizing inside profusely. Sometimes, if you slice them, you could see 100% of the cells inside apart from the outer layer is colonized by the fungus. That means it is a compatible fungus. Otherwise, the orchid seed wouldn't let the fungus in. So you can see the blob here. This is a magnified view. You can see the fungus here, and these are the seedlings. Fungus forms a mat over it, and then it germinates, and that is a compatible fungus. And these are the stages. Sometimes it gets to this stage, then don't go beyond. That means that is not a compatible one. You have to reach this stage. That is the stage where you can see the shoot pole, and then profusely producing rhizoids and the root. You may be able to note here. So that is a successful symbiotic germination. So there are so many, additives you add for as I said earlier, with the asymbiotic seed germination. In the case of Cyridedium calceolus, you need to give all the nutrients it needs, and for the symbiotic seed germination, you don't need to give anything, just oatmeal agar and then the mycorrhizal fungus. So that will do the job. And at the end, this is a successful symbiotic seed germination. It is profusely the rhizoids. These are the first roots, and then the primary taproot. It will come, and then you got a symbiotic seed germination. So how the orchid will let the symbiotic, the compatible one in. So if I go back to the other slide, you can see these rhizoids. So if we enhance this magnified view showing the fungus is going in through here, and they are colonizing inside. But if you look here, the incompatible incompatible fungi will, languishing around the orchid. It doesn't get in. So there is a signaling mechanism which allows the orchid to understand or identify which is a compatible mycorrhizal fungus. So I just to give you a very quick update of what we have done in Madagascar with all this information. There are nearly 1,000 orchids in Madagascar. Of these, 90% of them are endemic. So there's a serious problem in Madagascar because they use this slash and burn agriculture, illegal mining, charcoal production, collecting orchids from the wild. So this all contributing to the decline of orchids in the wild. And as you can see, this must have been hundreds of years ago, must have been a pristine forest. But what you see now is just, rice fields and people's other crops are growing in these. Cats sitting there on a large boulder, really large boulder, collecting some orchid seeds. So when we went there, we could see amazing diversity of orchids, but at the same time, the habitats are so, sort of fragmented. And the perfect example is this one. This tree is almost to, about to die maybe because it hasn't got any leaves or anything, but it is a haven for these bulbophyllum orchids. And these are orchids which are found on boulders, and how much organic matter is there? It's just a bit of lichens and some organic matter. So these are orchids living on the edge there. So it's really amazing place to see orchids and to do conservation. And some of them are very habitat specific, like this cyanochis. You can see it is by a stream. It needs moisture, most of the year around. It needs moisture, otherwise they won't survive. And John, my colleague from Kew, is listening here, and he once told me that this area, I think I am right, this area recently been mined for marble and granite, but at the same time there was a fire for charcoal collecting. And so there are so many problems in Madagascar where for an orchid to survive, it is a challenge. So these are some of the orchids we studied. The reason I am putting this list here is these epiphytes, I said epiphytes and lithophytes are not vulnerable to extinction as the terrestrials. But in a country like a biodiversity hotspot like Madagascar, they are prone to extinction because of the other challenges. Populations are fragmented and the other challenges it face. And what we have done, as I earlier showed, we produce symbiotic seedlings and we send these symbiotic seedlings back to wild. This is Cyanochis purpurea. So if you slice these roots, you can see the mycorrhizal fungus inside. So that means this is the perfect propagule for going back to the wild. That is the same case with this Angraecum protensum. This is a lithophyte. But as you can see, there may be an orchid here. But actually, this should go into a rock surface with very little moisture. So at the time that my team went there with a a filter, a very tiny filter, it lets water in, water out. So they made an impromptu watering system. It lasts for a few months. 2-3 liters of water and a filter. It drip irrigated these tiny seedlings. Otherwise, they won't survive during the summer months. So that is possible, even if it is a harsh, climate like in the central highlands of Madagascar, it is quite dry. So the Kew Natural England partnership, nearly 30 years of work, as Glenn earlier said, we supported the reintroduction, reinforcement, and that sort of work, which with the asymbiotic process. At the moment, the focus is identifying the compatible mycorrhizal fungi and for reinforcement, reintroduction work in quite a lot of other species. This is one of the species which we are working. We've done some work in Loire Valley, France, because we don't have many population left in UK to collect root material. We went to France and collected some materials. One thing we straight away identified was, these plants were growing in a very healthy environment where they produce so many flowers. Some of them look really fantastic. And once we came back and done some, sorry for this clumsy graph, but if you just concentrate on this red writing, what you are seeing is they are all mycorrhiza. This is the left-hand side is the pH is just below 8, and the phosphorus content is a bit high. And here the pH is above 8 and very low phosphorus content. As you can see, the percentage of mycorrhiza is quite low. The density is quite low. Distribution is there. But on the right-hand side, if you look, these red letters, they are all mycorrhiza. They are quite high. But at the same time, if you look here, it is so crowded. Actually, the orchid is recruiting all sorts of fungi, thinking some of it will come and help us. So basically, it is recruiting everything. As you can see, so many fungi here. But the one with the mycorrhiza, it doesn't need to recruit that many fungi, which are non-mycorrhizal. It is recruiting less fungi. And when we compare these plants, this group of plants were far better than the ones here. So that tells you a lot about the contribution the mycorrhiza made even after the plant is, the seed is germinated and it is established in its natural habitat. Then Cypripedium, I'm not going through this. This is the plant in the wild. Too much detail, but this is what we do. As I said earlier, we get the seed capsule, we germinate it in vitro, and then the maturity of the capsule, this is a sort of empirical sort of number of days, 45 to 60 days of maturity, you will get the capsule ready for in vitro germination. But if it becomes brown like this, it doesn't germinate. Then it needs a mycorrhizal fungus. At the moment, we haven't identified that. And as here, Galena is taking out the seedlings and bagging it and then put it in the chiller. After a couple of months, you can send it to the volunteers and they will look after it up to the point where they can be reintroduced back to the wild. And these are the reintroduced plants. Glenn's pictures was amazing, some of these plants in gate barrows. So continuing on with the theme, what I'm trying to say was the mycorrhiza is important. The man orchid, we got populations in Essex Wildlife Site, and I and my team went there 5 years ago. We collected some samples, hoping that we may be able to identify the mycorrhizal fungus. We were right. Within a year and a half, we got the mycorrhizal fungus, and then started seed germination after a few years, and now we got very successful seed germination. If you want to reintroduce or reinforce these seedlings back to the wild, it is possible. The next one with the Natural England funding we are doing is the early marsh. Only 10 plants left in the wild in Suffolk. It is, as you can see, it is caged. After the pollination, you don't really want to lose these seeds. And we collect these seeds when they are mature, brown seeds, and they bring back to the lab. And then the problem is, I need to go back to the slide. So they won't allow us to get any root material from this because the problem is, if we get any seed material, root material from this, then the plant will die. If sometimes it may not happen, but for our purpose, we need quite a lot of material sometimes. So we were not allowed to touch these plants. So what is the next approach? So what we have done is, So what we have done is, we went to Essex, and they got really successful colonies of different species of dactylorhiza. So these orchids are in different habitats. So we collected quite a lot of orchids, and we tested these early marsh orchids, hundreds of different lineages of mycorrhizal fungi with these yellow early marsh seeds. Luckily, just only one lineage was useful for the seed germination, and we got the successful seed germination, and we produced hundreds of seedlings. We done all sorts of testing. Glenn was earlier saying about climate change. We even tested for sort of prolonged drought. Is sort of we simulated inside the growth chamber whether you can give them very little moisture and then a high temperature to see whether these orchids would survive. They survived. I think it is just because they got the right compatible mycorrhiza inside. The seedlings we produced without the mycorrhiza, they lasted only for a couple of months, but they never grew to this size at all. So that means these symbiotic seedlings can be put them back to the wild for reinforcement studies. So with quite a lot of trials and things, what we have done is we collected soil from the wild, brought back to the lab, and then mixed with our own mix and see whether we can germinate, we can grow them with the wild soil. That is a test. That is a litmus test, because the wild soil got quite a lot of other fungi, and if we put these seedlings, whether those fungi will sort of colonize inside and then kill off the seedling. But that didn't happen. They grew really well, and a couple of months ago we reintroduced back these seedlings in Suffolk, in another area, not in the same place. And these are tiny. John sitting there, taking notes. It was a really fantastic day there, because we had something nearly 100 seedlings. As you can see here, this is the type of seedling we took. It's so tiny, but this is a scale. I think we got a really I would say 15 centimeter long. So we had a 10 centimeter long, may not be 10 centimeter, maybe 6-7 centimeter long seedling. But the problem is it is a fen area. So if there is quite a lot of flooding happening, then this may disappear. But we are hoping it doesn't happen. As you can see, one plant gone into that, and there won't be much soil. There isn't. As you can see, this is a fen, because quite a lot of the top soil is of mat of roots and these reeds. So there isn't anything. So to push these plants into the the soil was quite a challenge. We really hope, fingers crossed, that some of these plants would survive. So that was a successful, sort of from seed to the plant to the stage of reintroducing back to the wild. Then comes the green wing orchid conservation program, and so the identification of mycorrhizal fungus was the challenge. Basically, we initially thought we go there and collect the the root material, and we had a plan mapped out with Stephen, and we almost, I think we booked even the hotel. Then came the COVID-19, so we changed everything, and then we had a plan to get the roots from the wild side sent over, and then we will try to do that. That is not the best approach though, but that is what we decided. Then for the seed germination, the right maturity of the seeds is what is required. So Stephen had a fantastic volunteer come enthusiast, and she really helped us to get the right maturity of these seed pods for the asymbiotic seed germination. That means without the fungus. That happened in May, June time. Then to reintroduce back to the wild, this is a long process. So we need to compare these asymbiotic and the symbiotic seedlings. Now, I can say that the results are really encouraging. So these seed capsules we received, some of them are at the right maturity, optimum maturity. We got some seeds. They germinated, as you can see here. Some of them go to the green bit at the tip. That is the seed. The seedling is being developed, and some of them we transferred into jars. They are they are growing as successful seedlings, and hopefully we will get quite a few seedlings to try. So it is possible. Even if we had quite a lot of limitations, we worked around it. And I have to thank John here, because at the time it was a lockdown. We were not allowed to allowed in at Kew. So what we decided was the seeds will be sent over to John's house. So we developed a method 10 years ago, not 10 years ago, it is about 17 years ago at Kew, that even if we don't have an aseptic facility like a laminar flow cabinet for sterile air inside the lab system, there should be systems to grow these sort of very rare plants in a very, very primitive sort of setup. So on the kitchen bench, we got this medium, which is made up of some antimicrobial stuff. That means even if we open the jar, there won't be any fungus or bacteria growing on it. So that sort of medium we made and put the seeds in. So we were hoping nothing will grow, that happened, and we got this, the seedlings. So we don't do this for a project like a, Natural England project or Broom project when we started it, but the COVID sort of situation forced us to think plan B, and the plan B worked, to be honest with you, and hopefully we will get some seedlings. And for the symbiotic seed germination, Stephen sent us some really beautiful plants, and these plants, they were so good, and you can see very healthy roots, and we sliced these roots and we got successful colonization inside these cells. So we picked out these, we call these pelotons, these super coiled mycorrhizal fungi. And what we found out was this colonization is so good, so we could, grow them. And I was at the time hoping some of the things we were growing will be, sort of single mycorrhizal fungi. The reason I'm saying this is you may pick this peloton of a super coiled fungus and grow it. Sometimes you get different mycorrhizal fungi within it, and also other fungi. That can be a very, very difficult situation because we have to identify the right mycorrhizal fungi. But if we got so many other fungi, then it is very, very difficult. So, but in this case, it was only a single fungus, and we managed to grow these seedlings from the seeds, the mature seeds, or the brown seed pods. Stephen managed to collect and send to us. So what we are seeing here is the single mycorrhizal fungus compatible for seed germination. And what you may be able to hardly recognize, but you can figure out here, there is a shoot pole. That means this is a full germination. And if we slice these protocol seedlings, we may be able to see the mycorrhizal fungus inside. So basically, I would say there's an element of luck involved. I need to go back to this slide. So there are so many roots here. So if you slice this root and get this, and if you slice this one and get similar colonies, you may not get the same mycorrhizal fungus. As I said earlier, the French example, that is the reason why I put that slide there. There were so many mycorrhizal lineages in there. And similarly, here, green-winged orchid also may be recruiting different mycorrhizal fungi. So if we slice all these roots, what you are getting will be different types of mycorrhiza. Some of them may be germination specific, but some of them may not be. So if that is the case, our job is quite difficult. But these single pelotons, these mycorrhizal fungi we collected, some of them were the right mycorrhizal fungi. So they germinated that means we got a symbiotic seed germination, or these seedlings are symbiotic. So in most of these cases, we get some of these, like in 10-20%, we get the perfect scenario where we can get all these, orchids, identify the mycorrhizas, symbiotically germinate them, then produce the seedlings, and can support, important conservation work as part of Boom and similar projects. But sometimes the viability of the seed is a serious issue. We come across with the red helibrine seeds in the UK. And if that is the case, then symbiotic seed germination can be a real challenge. Then small number of plants, as in the case of Cyridedium, means we can't collect any mycorrhizal fungus from that wild plant. So basically, that is a no. There is no way we can collect the actual mycorrhiza which is in there if it is still available. Then the identification of the mycorrhizal fungus becomes so difficult due to natural seedling recruitment. So, for example, in the case of Cyridedium, there is a clump, a very successful plant there, but there isn't any mycorrhizal fungus mediated natural recruitment. If that is happening, we can get some seedling material and then collect it. Then seed germination specific mycorrhiza may not be culturable. Say, for example, we done a very large trial for the red helibrine for the last 2 years. We had mycorrhiza from the Lua Valley, but none of them were germination specific. So that was quite a setback. And now what I'm doing with my Czech partners is to bait the seeds. So they baited seeds 18 months ago. We got the first batch of seeds came back to us 3 weeks ago. We looked at nearly 5,000 seeds and none of them germinated. That means 18 months gone, nothing happened. So that means you have to wait for another 18 months to see whether we will get the germination specific mycorrhizal. So these are some of the bottlenecks. As I said, this is a viability staining method we use. This stain shows this is the only viable seed here. On the right hand side, this is the germination in vitro in petri dishes with all the nutrients given. Look, all the seeds sitting there, but there are only 2 of them germinating. Similar case, this is a cyanochis from Madagascar. This is an Aranchis from Madagascar. Here it is a staining and here it is the culture. Only 1 seed germinated out of 20 or 25 seeds you can see there. So that low seed viability is another issue with some of these orchids. So these are the challenges when even if you can identify the mycorrhiza. We got mycorrhiza from here, but here this is a very good success story. I think this is one of the major stories from Madagascar. What we found out was this orchid, this is called Aranchis ellisii. The seeds can be germinated by a narrow lineage of Cyridedium fungus. And we published this recently. And that means this orchid species is prone to extinction if the natural habitat is more fragmented. So these are some of the challenges. There are other things. It's a bit dramatic. I just want to show you, you make all these plans. We made the plans. John helped doing the planning, get the permits, all the things. We know where we are going. We got the team in Madagascar ready. We got nearly 6 of us going from UK and the US, our partners from the Illinois College. We went there. And the next day they took the van and with all the materials for the trip. Halfway mark, they found they can't go beyond that point because there are no bridges, all blown away, and the road is, as you can see here, is not there basically. So they had to change plan and they went somewhere else. And in that 2015, they collected some of these orchids, and we are publishing in the next few months. It was some of the amazing orchids we collected from Madagascar. So even if there were so many setbacks, sometimes in working in a biodiversity hotspot like Madagascar, you may not run out of ideas and places to go and collect your orchids. So orchids are so special, from our own country and from these places. And one of the areas which I am working at the moment is to find out ways how we can use orchids for other things, to connect people with nature. This project is all about that. And one of the things about orchids, they are so beautiful. So there is no introduction needed for an orchid to be, to shown to people, and then the impact it makes. I want to give you a couple of examples. This is a bit out of the the context of the work, but look at what is done in Singapore. It is a city state, but the city is full of orchids. They produce these orchids. Some of these are native orchids, and they produce these orchids and put them on trees, epiphytes. Most of these are. So for people living in the cities, it is one of these, one of the most iconic plants they can see when they visit city. In the UK, some of these inner city children, especially the young generation, won't see many of these our wild plants and countrysides. So orchids are quite suitable for that sort of work. And this is a very sort of unique project the Fairchild is doing. This is called a Million Orchid Project. I want to show you just because with the help of university, school children, senior citizens, they're producing 1 million orchid, put them back to Miami. Miami is a huge city. The South Florida native orchids will be in Miami. So even if these children or young people, the senior citizen can't visit these places, or the people visiting the city can see. So orchids are really good, charismatic plants to connect with nature. And this project is all about connecting with nature. I'm almost there. Got to a couple of minutes. So orchids improve landscapes for beauty, its charismatic nature, and also it also tells about the soil condition. So I didn't show much soil data, but the 2 slides you saw, the soil is the important part. So we collected some of the soil from the wild site, which Stephen sent to us, and these seedlings will be grown in the lab with the soil to see what sort of soil conditions needed, when they have to be, for assisted colonization work or translocation work we need to do in 2 years' time. So they are so amazing, but at the same time, it comes with quite a lot of challenges. And with the boom work, I think we were so fortunate. I wouldn't say it is our methods. I think we were quite fortunate to get these things working during this very difficult time. Even if we were at home most of the time, we managed to get quite a few things done. And hopefully in the next few months, once we are fully back, we may be able to do this work in full swing. So I have to thank these many people. I must have missed so many people here. There were so many, really brilliant scientists, technicians, and quite a lot of partners from other countries. I can't thank enough all these people. There are some amazing students, Sandwich students. They come from universities and spend 1 year with us. Then there were some interns and volunteers. Then the funders, Natural England mainly, then Sainsbury, Orchid, and there are so many funders. And they, even if it's a small amount, it really helped. And also, the partners which we are working with, UK Native Seed Hub from Kew and Plant Life and some other agencies. So it was, I would say logistics is one of the main things with this work. Sometimes getting the right stage orchid seeds, the best quality root material, and for that we need an army of volunteers and helpers to get these things done. So I can't thank enough to them. So thank you, all of them. And so I'll be happy to take any questions you may have. And thank you.
Wow, thank you so much. That was an incredible journey there into all the work you've been doing, Sarasan. Really appreciate that. I have a feeling there'll always be room for charismatic species within conservation world, and it's really, really interesting to see what lies behind these things. So yeah, really fascinating to see like the work that's going on. That links what's going on in South Cumbria with what's going on in Madagascar and France and everything. So massive thank you for that journey there through the work you're doing. Really, really interesting.
So, hi everyone. This has changed a little bit because unfortunately our Zoom recording ceased just after Dr. Sarasan's talk there. So this has been added on a little bit afterwards. This is just to give you a little bit more information on Boom, what we're doing, what we have been doing, and also how to connect with us and get involved if you want to. I will do a little bit around the actual plants themselves. I know our speakers have covered them and had some pictures of them and described them in some detail, but I'll quickly go over a little bit more information around them. The flower itself is quite a short orchid, 7 to 15 centimeters as usual. However, it's not that strange to find ones much larger than that, and certainly where I've been working on the lots this year, 30 centimeters wasn't an unusually large flower in some of the lusher areas. Late spring is its traditional flowering time, and certainly where we were working this year, May, and just into June was when we found them. However, by the time they had got to June, they really had started to go over. The flowers are pink to purple, occasionally white, and indeed most of these colours were found on the lots this year. A little patch of white found there as well. Now, the green-winged part of the name has been described by Naomi as to how it gave rise to the Latin name, but it's very clear if you look at the plant also where it comes from. And these stripes on the hood really are quite distinctive and stay visible on the flowers up until they go over as well. So you can still see this distinct striping on the sepals here. The leaves are thin and pointy, and there shouldn't be any spots when it's a young, healthy plant. In terms of habitat, certainly what gets quoted for most sources for this is unimproved grassland, and that matches what we're seeing here in the Morecambe Bay area, particularly coastal grasslands for us, the Lots, Jack's Gout, Humphrey Head, they're all coastal grasslands where we will find these species, but also coming into the hinterland, they're not unusual as well, and certainly pasture and open woodland have been mentioned here. Would be really important to mention, of course, that the unimproved part here is really important. And so when they say pasture and open woodland, it's likely to be good quality, robust open woodland, and in terms of pasture, unimproved, where you find it still. In terms of distribution, I've got a couple of maps here. The one on the left is a map of all records from the turn of the last century, so really going from 1900 all the way through to the current time. And as you can see, they really are distinctly focused in the southeast and central part of the UK, with fingers coming up the west coast of Wales and up the east coast of the UK as well. And once you get past the populations we have here in the Morecambe Bay area, there really is only one small population left on the west coast, and that's up in Scotland in Ayrshire. You move across to the right-hand map. This is from all records from 2000 onwards. And in terms of the range, it really hasn't changed very much. You can see still focused really in the south, east and central parts of the UK, with the same fingers going up the east and west coast. However, if you look within it, you can see the distribution really has contracted, and it's really not so dense in terms of where you find them. In the inset up there in the top right hand corner, that's the Morecambe Bay area, and if you compare that to the inset on the left hand map, you can see that the same thing has happened again. The many sightings across a large area of their distribution, really contracting to one or two key sites that keep on coming up year after year. We have talked a little bit around this, and it's the usual culprit in terms of grassland species, and it's all to do with agriculture, both improvement through the 20th century and the intensification of agriculture, particularly the addition of fertilizers, which have a huge impact upon the soil mycorrhizae, and as you see from Dr. Sarasan's talk, they are all important to the well-being of these orchids. But also just the changing of practices and the ever-changing guidelines and structure around how subsidies work. These change a lot, different management ideas come out. It's going to have quite an impact on particularly shorter orchids like this, plants like this. You can't talk about threats to these plants without talking about the ever-present spectre of climate change. This has been discussed already. I put this picture up on the right here because I think it kind of gives a good indication of what we might be looking to expect. This is actually the Lots this year, and this is mid-May, and as you can see, the bank here where the soils are at their thinnest has become incredibly dry. This was during that really dry period we had. I think it was around 5 or 6 weeks of really minimal rain, and what you can see here is this bank started off full of plants, lots of flowering plants, and then quickly they went over and sort of died back and were very difficult to find at the time of year when we were looking for seed. Down in the lusher area in the bottom here where the soil's deeper, you can see the grass is lusher, greener, and much more robust plants were found down there in terms of the orchid as well. And I think this might be a microcosm what we're really looking at. These thinner soils, which are marginal, but okay, so long as they're getting the moisture within them, are really going to become problematic if we do experience long dry periods during the real growth stage of these plants when they're trying to gather energy to set seed. So it might be that these plants become sort of really pushed into these areas here, not unlike what Glenn was talking about with the lady's slipper orchid, where moist areas around Haweswater were found to be much more sustaining for these rather than the marginal areas on the pavement. And this all leads to the story, as you see at the bottom here, that this plant is now globally listed as near threatened on the IUCN Red List, and that same designation is mirrored in the UK Red List as compiled by BSBI. So what have we been doing? Well, all the way through 2020, we had big plans to lots of engagement and lots of survey work, but obviously coronavirus has had a huge impact on not just us, but all sort of aspects of this kind of sector work. However, we did have a really important piece of work to do at the beginning, which was working with National Trust, who own and operate the lots. A methodology. We did that through their National Environment Advisory Board, and they really helped us. We liaised with Naomi on that and really helped us put together a robust scientific-based methodology for collecting seed and material on these plants so that the project could pursue what its aims were, but also the area that we were working with was protected for future generations of plants. And that was really important, and that worked really well. I'm really thankful to the NEAB for all the advice and the feedback we got on the application, and it was approved, and that's why we've been able to do this work this year. With the monitoring of orchids, we had a big plan, like I said. They were really put under a lot of threat by the shutting down of travel and everything like that. We were incredibly lucky, as we've mentioned before, to have Julia Sire, a local woman who works for the Cumbria Wildlife Trust, but has also been incredibly active in volunteering and helping the project. And she was local enough in that she could take in the lots as part of her daily exercise during lockdown. So she was able to traverse the lots on a number of occasions, keep an eye on these plants for us, and she was the one who fed back through pictures that these plants really were maturing and then going over quickly this year. So I was forwarding these onto Kew, and the decision was made that we had to move quickly to get this seed, which we did, and we took them over a number of weeks, different maturities, and passed these down to Kew, and they were able to find enough viable seed at the different stages of maturity that were required. So we can't thank Julia Sire enough for the efforts that she put into making this happen, because really we probably would have potentially missed any seed this year if we'd just been looking at traditional seeding times for this species. So that on the ground observation was essential, and yeah, really has enabled us to kind of keep on track with this project. The root material was supposed to be collected by Kew early in the year. Obviously that didn't happen. We did try to keep on putting off so that Kew could come and do in as sterile and sort of non-invasive way as possible. However, it got to the point where we really did need to achieve what we need, our targets for this year. And so as me and Julia Sire again went out to collect the root material. And as you see on the right hand picture there, that was a core of soil we actually took. I had marked plants earlier on in the year just in case this did happen. So I was able to return to site, find my markers, know exactly where these plants were, even though their above surface part had died right back, and take these cores, send them down to Kew, and they were able to extract the plant's root system, as you saw in Sarasan's presentation there. And yeah, really has kept us on track this year. I'm really happy to see what Sarasan said in terms of progress. So yeah, can't say thank you enough to Julia, but also just to point out that a lot of beautiful pictures on my presentation here. Julia again was probably responsible for most of those, and a gentleman called Tom Crawford, a local amateur photographer who was out on site, met Julia one day, we were put in touch, and he's passed on some pictures as well. And some of the gorgeous pictures you'll see here are thanks to those two individuals. So quickly get that in there to say thank you. What are we going to carry on doing? Really, it's not stopped. We've been doing this since the very beginning, and it's building up that partnership, building up the stakeholder groups, and really just trying to reach out and talk to as many people as we possibly can. Really need to mention Cumbria Wildlife Trust here. We have been in quite lengthy, detailed conversations with Cumbria Wildlife Trust around sites that they own and manage. Humphrey Head springs to mind as an obvious one because there are populations of green-winged orchid there. But we need to continue having those conversations. We talked last year to Arnside and Silverdale AONB Trust, and then through them we were put in touch with their conservation grazer, who talked about the fact that an area of land they had taken over very recently, and only a couple of years ago, that they'd put onto a slightly different grazing regime. A green-winged orchid had come back in that field where they hadn't been recorded before. That's massively interesting for us in terms of the project and deciding what our direction is going forward. It gives the sign that there may be extant seed banks in the soil, and that these are still viable, and that actually, you know, there are still very specific management regimes that will facilitate the comeback of these, but also just points out there's very specific management needs for these to thrive. So working with that organisation and that grazier and wider areas and landowners who may have these, start trying to build up a really clear image of what these plants need, what makes them thrive beyond the obvious mycorrhiza and unimproved status, but also just what grazing is doing them the biggest service. And we'll need that to inform how we look at recipient sites in the future, because if those things aren't in place in terms of soil health, but also ongoing management, then we really are going to be sort of not be able to achieve what we need to. Yeah, we have been trying to establish a monitoring of populations, and really that's been about talking to the organisations already who monitor these places. Cumbria Wildlife Trust and National Trust already do an incredible job of monitoring their populations, and we'll really just be looking to add resource to that if required, but also to learn from their methodologies and try and standardise it, but ensure also that it can feed into important national scale research if necessary. So what's to come? Really a continuation of what I've just been talking about, but a lot more practical alongside that. So as well as talking about and looking at the survey methodologies, we'll also actually be getting on the ground and starting to do some, and that hopefully will be involving members of the community who are interested in learning those skills. We do need to identify feasible donor sites. We were focused on one site this year, that's the Lots, and we were incredibly lucky, and it is the best site for collecting this material from. But in terms of going forward, we would like to look at some of the genetics and the DNA work on these with Kew. So we will be looking at taking material from other populations in the area as well. So that would mean moving over to some of the Cumbria Wildlife Trust sites where they have these plants and looking at whether it's their feasible donor site, i.e. is there enough plants, does their future look secure enough for us to start taking material from them. But also start to identify feasible recipient sites. This is looking at things I was talking about in the previous slide around what makes a site suitable now, but also into the future. So we really need to start putting together a list of sites and landowners that we can work with to start putting these plants back into the area. We will continue to liaise with Kew. Their work is not done in any way. Really good start to the work this year, but we need to continue with that. We will be continuing to send them seed down. We will also receive seedlings back from them at some point. As soon as Kew are happy that they've gotten to a stage where they are robust enough to leave their hyper safe environment, we will be taking those seedlings back, but we will need to continue to look after them. They won't be ready to plant out for an unknown at this point number of years. It may well be that we need to look after them for 2 or 3 more years. That means we'll need people to help us look after them. The individuals on the project team could look after some of them, but really this is about community, and we're looking for people to come forward, put their hands up and say, yeah, I'd really like to get involved in this. I'd like to look after these plants. I'd like to sort of take responsibility for this part of the project and the next part, which will be planting them out. And to assist in that, we will be offering training and support and resource all the way through it. This is a really important part of it. As you've seen, this is an incredibly complex procedure, a lot of time, effort and money put into it, and there's a chance that that might actually put people off and they might be a little bit daunted by it, but it's really important to Boom that we actually let people know that this isn't just going to be right, these things, look after them. There will be a lot of input, a lot of training, a lot of support. You will be able to feed back to us and to Kew, and there will be real sharing of knowledge, and we really will be trying to bring those people who agree to give us their time and effort to look after these. We really will be looking to support them and develop them in any way that they think is needed to help us do this. As I said, collecting seeds for Kew, I'll definitely be doing. But also there's going to be the wider engagement work as well. And that is going to be really about getting people out to see these plants, to see what we're doing on the ground. There's loads and loads and loads of people in the local area and the region who are really knowledgeable about these plants. There's also loads and loads and loads of people who have little idea about these plants, little idea about how amazing and unique and important they are. And we'll be looking through Boom to kind of really talk about that story and spread it wider and across the diversity of social factors that you have in this region. Like I said at the very beginning, we'll be looking to unify the diversity of society and nature and try and combine it into one sort of holistic package through Boom. So there'll be walks, there'll be talks, and there will be training and workshops as well. So that's it. Unfortunately, we did have questions at this part, and during the talk we did actually have some really good questions, and the panel were able to give some really, really interesting answers. Unfortunately, the recording did not work past that, and we haven't got them. I do have these questions written down, and so I will try to summarize the answers given by the panel to them, and that will be available to download as well. Okay, thank you very much. I am going to pass over to Michelle now, who has also put together a very short recording to discuss with you how you can get in touch with us and how you can get involved. And just say thank you again for everyone who attended the talk, all of the speakers and everyone who posed questions to the panel. They're really interesting, and really appreciate you giving your time to come and spend some time with Boom. Thank you very much.
Yes, so thank you. So Morecambe Bay Partnership is supporting the community engagement and volunteering strands of the Back on Our Map project. For anyone who doesn't know Morecambe Bay Partnership, we're a charity working all over Morecambe Bay, both in Cumbria and Lancashire. And we work extensively with community groups, local organisations, volunteers, and anybody really to collaborate to make good things happen around Morecambe Bay. And it's our aim really to conserve, celebrate the really rich cultural and natural heritage of the Bay. And we also do a lot of work around access too. So you might know us for the Bay Cycleway. You might know us for some of our work around disabled access and improving routes into the landscape too. So we wear a lot of different hats, and all of our projects have people at the heart of them. And as Stephen said right at the very beginning, it's really vital for the Boom project that we connect these species with people, and we engage as many people as we can through really high quality events and volunteering opportunities. We want to be able to share skills and then get people sharing those skills further with other people. And ultimately, I suppose we're trying to find communities and individuals who will be ambassadors for these species in the long term after the life of the Boom project. It's also really important to Boom that we provide opportunities for other communities, perhaps in areas where people don't get the same chance to get out into the landscape, and where people haven't had the opportunity to work with nature or experience nature and wildlife before. So just fostering a general interest in nature and how we can help protect it for the future is a really important part of Boom as well. So if you feel you can help with anything, if you'd like to volunteer, if you'd like to sign up to the newsletter, or if you'd just like to come to some of the events or just keep on top of what's happening with the project, these are all the different ways you can do it. This on the screen there. You can sign up as a volunteer via the Morecambe Bay Partnership website. If you just go to the Get Involved section, you'll find information there about our nature volunteer programmes, including Boom. You can just send us an email. You can follow us on social media. That would be brilliant. And do message us on social media too if you have any questions. That's my email address, or you can email the team on the boom@cumbria.ac.uk email address. And yeah, keep in touch, and we'd love to hear from you. And if you can all go away and tell 5 other people about the project and how they can get involved, that would be really fantastic. Thank you very much. Back to you.
So thank you, Michelle, for those short words about how to get in touch. I really hope everyone will. There was some talk during the actual talk today about the idea that if everyone who was at this talk goes away and tells 5 to 10 more people about the Boom project and about what we're doing, that will really help us to reach out into the area, the region, and even nationally, because I know we really were represented by people from all over the country on this talk. And we really want to hear about people who are doing similar work. Really important to the project that we learn from the existing knowledge base out there and build upon all the amazing work that's already been done. So yeah, thank you very, very, very much for coming and joining us today. If you have any questions, please use the contact details as given by Michelle and get in touch with us, and we really look forward to hearing from you. Thank you, everyone. Bye now.